Hello and welcome to Chapter 14. This is the Introduction to Carbohydrates, the first chapter in our series of biochemistry chapters. So if we take a look, we're just going to go over our overview really quickly. So we will talk about carbohydrates and their function. We will talk about we call stereoisomers. Iso meaning same, mer meaning same compound, but just how they look like in three dimensions. We will talk about what we call monosaccharides, mono meaning one, so what happens when we have one sugar. Then we will talk about disaccharides and polysaccharides. So di meaning two, so we have two sugars. How are they connected together? Then we talk about polysaccharides. Poly meaning many, so what happens when we put many sugars together? We will talk about how to name these, what their structures are, what their functions are, etc. And then we'll talk finally, the last thing in this chapter is using how we use sugars to define whether you have A blood group or B blood group or O blood group. So let's get started looking at the chemical composition of carbohydrates. So if we take a look at carbohydrates, or for short, we will call them sugars, right? All sugars have the same empirical formula. So the empirical formula for sugars are always, remember the lowest whole number ratio is what an empirical formula is CH2O. So for example, if you know that you have 6 carbons in a particular sugar, then you know the entire formula of that particular sugar. It would be C6H12O6. For example, glucose, you may have heard of glucose before, or glycolysis. Glucose is an example of a sugar with 6 carbons, and its chemical formula is C6H12O6. You may have also heard of a sugar called fructose. So if you notice, there's kind of a common theme here with the suffix of glucose and fructose. They both end with ose. So we know that for a particular sugar, if you see a suffix of ose, O-S-E, you know that it is a sugar. So if you take a look though, what you can see here to the right on the slides is we have something called a ketohexose. Okay? These are two important terms that we need to look at that define a sugar. So if you notice, we can look at the number of carbons that we have. So if you remember from organic chemistry, we have those prefixes like TRI, tri, tetros, or tetra. You may remember pentos for 5, or pent for 5, hex for 6. Well, if you take all of these and you add the suffix ose, that tells you how many carbons are in that particular sugar. So for example, here on the slides, you can see a ketohexose, hexose, okay? So here, hexose, this means that we have 6 carbons. Now, so therefore there, since we know we have 6 carbons, we know that the full chemical formula is therefore C6H12O6. If we have a pentose sugar, for example, says that we have 5 carbons, so we know the entire chemical formula therefore would be C5H10O5. And you could do the same thing if you look at the bottom with these aldotrioses. So tri means 3, so we have 3 carbons. So if we have a triose, therefore we would have C3H6O3. Now, you have a lot of different combinations that you can have for sugars because you can have 3 carbons, 4 carbons, 5 carbons, 6 carbons, etc. But another difference that you can have are these different functional groups. So if you notice with the top structure that we have on this slide, we have a ketohexose. The keto, so are we okay, this ending part here, hexose, this looks at the number of carbons. The keto here tells us what functional group do we have. So the functional group for a ketose is a ketone. And if you remember, a ketone is a C double bond O with 2 either the same or different R groups. We also can have an aldose sugar. An aldose sugar is a sugar with an aldehyde, which is a C double bond O, 1 R group and 1 hydrogen. So you should be able to, based on this information, if you're given, hey, this is a ketose or an aldose, you should be able to identify if a sugar is a ketose or an aldose. And you should be able to identify, is it a triose, a tetros, a pentose, a hexose, and be able to give the chemical composition based on that. Now, here's the thing that we're going to talk about next, is we're going to spend a little bit of time talking about the stereochemistry of these sugars. And that's really important because if you change the positions, like for example in this ketose sugar here, if you were to take carbon number 4 and change that OH group from one side to the other, you would make a brand new sugar. So it's just a very, very small change. It doesn't change the chemical formula. It doesn't change the functional group, but changing those other groups in 3 dimensions gives us a brand new sugar. And that's actually very, very important because these tiny little changes can lead to huge differences in how we recognize the different sugars. So that gives us a lot of diversity that we have, which means that we can have a lot of different signals and a lot of different communication that utilize sugars as a result. So a couple examples, number 1, virus attachment and entry into cells. So you may have heard of like the flu virus, for example, that binds to a particular sugar on the outside surface of cells, but only particular types of sugars. So that's why you oftentimes will hear about certain species associated with the flu, like bird flu or swine flu, etc., because different species have different sugars on the outsides of different types of cells. You can also, again, get communication between cells, meaning I need more water or less water or more of this particular molecule, or I need to get rid of a particular molecule. That can be mediated by communication with signals that are bound to or somehow introduced by these different sugars that you have. And then finally, what we'll talk about at the very end of this chapter is looking at those blood groups. So literally the difference between an A blood group and a B blood group and an O blood group has to do with sugars on the outside of your red blood cells. So before we get into talking about the different blood groups and all this fun stuff, we're going to actually talk about a little bit more in depth about how we get different types of sugars. Even though glucose and fructose are both C6H12O6, they are very different. Fructose is found in fruits, for example. Glucose is what we use to break down in glycolysis. So we need to understand that difference in chemistry in 3 dimensions to look at this. All right, so let's take a look at how we actually draw sugars in biochemistry. So to make things a little bit easier, rather than drawing the skeletal structures that you may be familiar with from organic chemistry, we use in biochemistry here what we call Fischer projections. So again, when you have these monosaccharides, as you can see here with ribose, we have CHO. So again, this is a shorthand way of saying we have a carbon with 1 oxygen and 1 hydrogen. This is a shorthand way of saying this is an aldose. So if you take a look, you can draw straight down 1, 2, 3, and we can draw CH2OH, and we can draw ribose. So ribose here, when we draw these linear projections or these Fischer projections, we can take a look at the structure of D-ribose and we can say, well, this is how many carbons it has and this is what functional group it has. So ribose is an aldose because it has an aldehyde functional group, okay? If you were to draw fructose, for example, here now we have a carbon with a double bond to the rest of the molecule. This group right here, this is a ketose. So you should be able to recognize and distinguish between an aldose and a ketose. But if you take a look, we have these functional groups, but then we also have the numbers of carbons that we can have. So again, the number of carbons and therefore the total chemical composition. Now in the example of ribose right here, each of these little intersections is actually a carbon. So we don't draw the carbons on either side, which we'll get to in just a minute, but if you count these carbons, you have 1, 2, 3, 4, 5. So ribose is an aldose, but it is also a pentose. So again, given a particular sugar, you should be able to determine how many carbons does it have, and therefore is it a triose, a tetrose, a pentose, a hexose. You should also be able to determine is it a ketose or an aldose. Again, now the final thing that we're going to talk about is the overall structure and how we get this first letter here. So this letter is either D or L. These D and L correspond to what we call enantiomers. Now, enantiomers are basically a way that we look at 2 chemical species or 2 organic species in 3 dimensions. Okay, so we'll get a little introduction to that in just a minute, but first I just want to go through a little bit of what we're looking at here when we look at these Fischer projections, because these are going to be a brand new way of looking at a molecule. But I can guarantee you, Fisher projections are a lot easier to look at for sugars rather than trying to draw them in line structures like you may remember from organic chemistry chapters. So, and the reason why we use Fisher projections is it's a linear, so you have a line straight down, okay, structure of these particular molecules. So this helps us to actually identify what we call stereoisomers because you have this plane, this straight line going down. You have things either on the left-hand side or the right-hand side. Now I could move one of these OHs here and the H over here. So along this line, that is where we're looking at these different isomers that we can get as a result. So if we take a look at our example here on this particular slide, we have lactic acid or we have glucose. So if you take a look at glucose or D-glucose, for example, glucose is also an aldose, okay? So we're going to draw our CHO group up top, and then we're going to draw a line straight down for our Fisher projection. And if you notice with the Fisher projection, we have a straight line, but the ends of those lines, the top and the bottom, are the terminal carbons. So we have 1, 2, 3, 4, 5 carbons in this particular case. So if we need to draw D-glucose, and we know glucose is a hexose, in the bottom here we draw CH2OH, we already know we have 2 carbons. So if glucose has 6 carbons, we already have 2, we're going to draw just 4 intersecting lines because each intersection here is a carbon. And by definition for D-glucose, by the definition of glucose, we have the OH groups pointed to the left, right, left, left. So we can fill in the structure here to give us, by definition, the structure of what we call D-glucose. So now, that whole point that we're talking about of D-glucose, this D group here, which we'll talk about in just a minute, again, has to do, these are what are called enantiomers. Enantiomers, they can either be D or they can be L. And this has to do with how these particular molecules look in three-dimensional space. So this first has to do with something called chirality. Chirality is Greek for handedness, because when we look at enantiomers are defined by saying they are non-superimposable mirror images. Okay, I know that seems like a lot, but bear with me. So if you take your hands, and if you notice, they are mirror images of one another. If you put a mirror in front of this hand, it mirrors the other hand. Okay? But if you have your hands like this, you have a mirror here going down. However, they are what we call, they are non-superimposable. So if I take my 2 hands on top of one another, right, they don't impose over one another, but they are a mirror image. Okay? So in order to get that first, though, we have to take a look at what chirality is, because that actually depends upon, gives rise to enantiomers. So chirality is, by definition, what we're looking at for carbons. So we have what are called chiral carbons or chiral centers. I will go back and forth between those 2 particular names. Or I just may say it is chiral. So chirality always refers to a carbon. So by definition, a chiral center or a chiral carbon is a carbon with 4 different atoms or groups, whichever way you want to think about this, on it. So for example, if you have an aldehyde, I have C double bond O, H to an R group. This has 1 group, 2 bonds to an oxygen, and a bond to a hydrogen. This is not chiral. So whenever you have a double bond or a triple bond with a carbon, it will never be chiral. So, however, if we have 4 different groups, then that carbon is considered chiral. So for example, are we okay that this carbon right here is not chiral, right? Because it's got a double bond O. However, if we go down to this next carbon here, we have an H over here, we have an OH here, we have an aldehyde group here, and we have, I'm just going to write it as the rest of the molecule going down here is an R group. This R group is different than this aldehyde group. This is a C with an A, OH, H, H, OH, H, OH. So this group down here is different than just this group here. So this carbon right here, I'll just fill it in, is a chiral center or a chiral carbon. So you can go down the line and you can see this next group has an H to an OH. This has 2 carbons, one of which is an aldehyde. If this had 2 carbons, one of which was an aldehyde, they would not be chiral. But because this R group is different than this R group, this also is a chiral center. Finally down here, H, OH, R group number 1, which has 3 carbons, R group number 2, which has 2, those are different. That's another chiral center. Finally here, we have an OH, an H, a CH2OH, and then 4 carbons up there. So those are all different. So here, so in the structure of D-glucose, we have 4 chiral centers. And that's important. That means that we can move things around those chiral centers, right, without changing the chemistry. And when we do that, we can get new molecules. So if you look on the slides, for example, here to the left, you can take a look at how many chiral centers are present in these particular species. So you can see here in glucose, we already counted that we have 4 chiral centers. If we look at D-ribose, okay, how many chiral centers do we have? Well, remember the aldehyde is not chiral, but we do have a chiral center here, here, and here. Now this carbon is not chiral. This carbon is not chiral because there's 2 hydrogens bound here. So that means ribose has 3 chiral centers. Glucose has 4. And then finally, if you look at D-glyceraldehyde here, we only have 1 chiral center because this carbon has 2 hydrogens and this carbon has the aldehyde. So you should be able to, if you're given a particular sugar, you should be able to look at the linear projection. You should be able to determine, is it a ketose, an aldose, a pentose, etc. You should determine if it is an aldose or ketose. So again, number of carbons, number of function, or what type of functional group. And you should be able to determine how many chiral centers are present. So again, here's just the example that I was talking about your non-superimposable mirror image using your hands. That's what we're looking at. This is where we get the D and the L versions, okay? So enantiomers, what that means is that you would differ at every single chiral carbon, meaning you would move the H's and the OH's to the other side. So let's take a look at our D-ribose, for example, here, okay? Now, the D and the L are enantiomers. So if this here is D-ribose, if we were to draw L-ribose, all we need to do is change 2 of the groups, switch the left and the right on each of the chiral centers. So we can draw our straight line, and so we know that the top and the bottom carbons are not chiral, so they stay the same. We're going to draw CHO and then CH2OH. And we have 3 lines for 3 chiral centers, and all that we do now is we switch these 2 positions. We switch left from right and right from left. So here the OHs are always on the right. We are going to change them now to always be on the left. And if you do that, now you have drawn L-ribose, because if we made a mirror here, these would fold up so the OHs meet one another and the H's meet one another, but they are not superimposable. So that's what we mean when we're talking about chiral molecules and we're talking about enantiomers, D versus L, okay? Again, so we have enantiomers, that's the D versus the L. They differ at every single chiral center, so you have to identify the chiral center, and then you have to move the group to the other side in the Fisher projection. Left goes right, right goes left. If you do that for every single chiral center, you create an enantiomer. However, if I were to take D-ribose and only move this particular OH, I would only change 1 chiral center. I would therefore be a diastereomer. Okay, so diastereomers differ at 1, but not all, at least 1, but not all of the chiral centers. So for example, if I have 3 chiral centers, if I differ at all 3, I would be an enantiomer. But if I only change 2, whether it's these 2 or these 2, I am now a diastereomer. If I differ at 1, either this one, or this one, diastereomer. So there's only 2 enantiomers. There's only a pair, because you can only differ at all. There can be many different diastereomers depending on how many chiral centers you have.